So this video is on some examples of proteins, just to go through a few key examples so we understand how they work and why their structure is so important. So in the learning outcomes here, really the key thing is understanding that difference, that connection between structure and function, how proteins provide that for cells, but also some examples of their structure and function. And we're going to look at enzymes, hormones, receptors, and antibodies, and look at those. But there are many different types and functions of proteins, so you have a few different ones here. Really, we think of proteins as being the main molecule that does things in cells. So if you need to hold something together, move something around the place, recognize something, that's what proteins do. So some of those functions include catalyzing reactions, and that's what enzymes do. And that's really a huge class, because what's going on in cells is a lot of chemical reactions, but they tend to be quite slow if you just leave them to go on their own. The enzymes help those reactions happen faster, and because they control the speed of the reactions, they also control whether reactions happen or not. And this level of control is extremely important in cells. So for reactions to happen, enzymes need to be there most of the time, and they're controlling those reactions. Whole topic on enzymes coming up soon. Signaling, in terms of if a message needs to go from somewhere in the body to somewhere else, or somewhere in a cell to somewhere else, chemical signaling through through proteins is very important. Transport of things inside the cell, or movement within the cell. So when muscles work here, what that actually is proteins moving inside a muscle cell, and all of those muscle cells together will help to move your arm. So even things like running, that's about proteins moving tiny amounts at the cellular level. Storage, so here we're storing some iron, and structural support, either in terms of something external like this spider silk here, or the cytoskeleton within the cell is made of proteins. So in order to make up all these different shapes and functions, we're actually only talking about a few amino acids, 20 amino acids that can be put together in a range of different ways. I guess one analogy that you can use for thinking about this is thinking about a Lego set that has 20 different types of block. Some have a hinge on them, some are big, some are small, some are long, some are short. But if you put those together, 20 different blocks, you can put them into almost any shape and arrange different things. So the 20 amino acids are the building blocks of proteins, and the order, sequence and length of those polypeptide chains gives you an extraordinary range of folding properties to make different shapes for different proteins. And of course, because they have those side chains that have maybe a charge on them, or different binding properties, those chemical properties of the side chains also function. So the shape of the protein is really important, but then it might have also a sort of stickiness to it that helps it bind to something else because of those chemical properties. So understanding some key examples of proteins will help to illustrate that. So the key one, again, I talked about there was enzymes. So enzymes have a unique shape that allows them to bind the beginning molecules of a chemical reaction, which we call substrate, and then release the products of that reaction. So here in light blue is the enzyme. It's folded to create this cleft that's called an active site. That's where the substrate binds. And perhaps because as it binds, it folds a little bit and moves a little bit. Remember that the folding of a protein is not something fixed. It's determined by its environment as well. That could pull that substrate apart a little bit, weaken those bonds, and then split that into two products. So that's the way enzymes help reactions happen. A reaction is about making and breaking bonds, and the enzyme helps to make and break bonds. I also mentioned signaling. So here's an example of a hormone binding to its receptor. So through the body, this protein with this particular shape is a hormone that's being released. It'll go all throughout the body, but it only binds and causes a signal when it can bind to its complementary receptor protein. So here's the receptor protein on the plasma membrane. We have a binding event here. When that binding happens, another enzyme might be activated, causing a signal that goes into the cell, which has some sort of effect on cellular function. So signals can be carried around by hormones and recognized by receptors, both types of proteins. The last one here is then an antibody. So we heard of antibodies before, they're part of the immune system. They're also proteins. And so these proteins are released, so they're an extracellular protein outside of the cell, floating around in the blood, for example, or outside of the cells. And they're shown here by these sort of Y shapes. The role of antibodies is to bind to proteins, but not proteins that the human body has made, for example, not the host proteins, but proteins that attach to some sort of invader or pathogen. So here's a virus which is covered in proteins, or a bacterial cell which is covered in proteins. The antibodies will bind to those. So antibodies are designed to be sticky, like I said, and have complementary shapes that will just find anything that's not from the host, bind to that, and as it says here, they mark it. They also sort of clog it up a little bit from going around, and then immune cells of the immune system will come and consume, eat, destroy those bacteria and viruses and keep your healthy cells healthy. As I said, complementary shapes are a really important part of that, and there's a video there on complementary shapes that Mr. Lecornu's done. Thanks.